My name is Iyanna Tuyay, and my mentor was Dr. Liz McCullough. And I did a literature review on integrations of auditory regions leading to auditory processing and autism. What is autism? Autism spectrum disorder is a neuropsychiatric development disorder and is characterized by a distinctive pattern of impairments in communication, social interaction, learning, and behavior. Individuals with ASD often experience restricted or repetitive behavioral patterns as well as reliance and interest. In my research, I aim to account for the cortical/brainstem usage, neurophysiology, behavior, and touch base on auditory evoked potentials, and N1Cs, which are a haplogroup, Y chromosome DNA, and brain hemisphere dominance factors that play a role in auditory processing deficits in autism. And I also aim to find relationships between brainstem and cortical responses to speech-like stimuli, such as pitch, tone bursts, music, etc., in a literature review. And this is important because autism has frequently been associated with abnormal behavioral responses to auditory stimuli, which raises questions regarding auditory processing deficits and issues in ASD. I aim to account for the brain mechanisms and biology for such deficits. The first subject I would talk about are the dominants in brain hemispheres, and using an electrophysiological method, which are HZ tone burst stimuli, Brunet and colleagues compared the relations between auditory evoked potentials and verbal nonverbal abilities in children with autism, comparing that to children without. And their study concluded that the front central Y-DNA waves in children with autism actually didn't differ from their control group, and the Y-DNA amplitude was found only in the right temporal site in children with autism, with a lack of variation in the left side, and an atypical pattern of asymmetry and right temporal dominance of AP, auditory evoked potentials, when stimuli increased, strongly suggests a preferential activation of the right hemisphere in the brain in autism. And then in abnormal cortical processing in the brain, in a similar review, Bodert and colleagues investigated whether abnormal cortical processing was also prevalent in children with autism, as less activation of left speech-related temporal areas in adults with autism is. Researchers ran a position emission tomography auditory activation study in children with autism using speech-like stimuli and found that between the first and control group, there was significantly less activation in left speech-related areas of the brain in the autistic group. And the image to our right shows abnormal activation of left speech-related brain areas, with the left middle temporal gyrus and precentral frontal gyrus were significantly less activated in autistic children. The second topic will be about cortical and brainstem usage. And brainstem and cortical responses to speech stimuli are the most important findings when using neurophysiological methods and data processing to make relations to language impairments and auditory processing deficits. Russo and colleagues conducted a study analyzing brainstem and cortical responses to speech stimuli from children who've completed a training program that trains language through games. Results showed that children with prior training showed changes in brainstem and cortical response times, so hence like a faster brainstem response time. And through Russo and colleagues' study, it questions like the benefits of such programs. Like, do these types of programs result in a faster like cortical response time? The image to our left shows the brainstem timing change in response to the word da in their study. And then let's see. Regulation of auditory input in individuals with autism. In a second study, Brunet and colleagues investigated a late auditory evoked potentials in autistic children at the cortical level. The study was interesting because while there is much information on abnormalities in the ability to regulate auditory input in individuals with autism, not much evidence has been put into the systems responsible for such regulation. Auditory evoked potentials are present in higher proportions in autistic children, but are not necessarily a condition like that is required, since many autistic children display normal brainstem AEPs. On the other hand, data concerning Y-DNA in autistic individuals have never been reported, which is what this study aims to focus on. So researchers ran diagnoses with 3 groups of 16 children, controlled and those diagnosed with autism, and they diagnosed them according to criteria defined in the Diagnostic and Statistical Manual of Mental Disorders, which was published by the American Psychiatric Association. Results found that intensity effect was prevalent in both control groups, whereas in children with autism, it was absent on the left side. And evidence in autistic children suggests dysfunction in brain areas involved with the Y-DNA generation. And then with my third topic, which are AAPs and N1C, which is the Y-DNA. So what are these 2 factors? Auditory evoked potentials assess specific areas of the brainstem, midbrain, and auditory cortices. It is an electrical signal evoked by the brain while an auditory stimulus is presented. Evoked potential tests, such as those tested in electrophysiological methods, record how quickly nerve signals reach your brain. N1Cs are a haplogroup N, which is Y chromosomal DNA. So in short terms, it has to do with the genetics and phylogeny, which is the branch of biology that deals with evolutionary development and diversification of a species or group of organisms. And with AEPs and Y-DNA, researchers Brunet and colleagues, which was previously mentioned, questioned the brainstem AEP abnormalities present in autistic children. Middle latency AEPs, which are the initial activation of the primary auditory cortex, for example, are normal in adults with autism, but have not been researched with autistic children. So what they found was an atypical pattern of right temporal dominance of AAP when stimulus was increased. So in conclusion, this paper is analyzing such auditory regions, methods, and stimuli, giving evidence for atypical processing of auditory information ASD, behavior-wise and neurophysiologically. And by identifying all these factors that contribute to auditory processing deficits, further research can be able to identify the underlying mechanisms that may cause or influence such deficiency. Thank you for following along, and I'd like to give a special thanks to my mentor, Dr. Liz McCullough, and the Talaria program directors.